through the corpus callosum all the way through the brainstem. So theoretically, I've got mirror images. Huh. Interesting. What do you guys see in this?
A big blood clot.
So there is a blood clot in the splenium of the corpus callosum, and probably one out in the genu of the corpus callosum. So in other words, the left and the right hemispheres probably weren't talking too well to each other, specifically for visual cortex as well as for prefrontal stuff. Okay?
That's better.
Can we do like,
Remember how I did the ambiance? Okay.
President for president.
Wow.
Oh, thank you.
It's way worse.
We can't see it. We can't do this one.
Too bright. Turn it on.
Just turn it off.
Sorry, Ari. Sorry, Ari.
Do we need to bring Steven Spielberg in this or what?
Wait, let me get my...
Yeah, I think so there.
Okay, so what I want you to look at in this first cut was, one, is there clearly two different pathologies in this brain, and it may be that I end up having to get a different brain once I start on this because of the blood clots. But what you can see on this is sort of that typical mid-sagittal view of the structures of the brain. So here's our corpus callosum. Then this area right here that's obscuring the lateral ventricle is called septum pellucidum. And on the other end of that septum pellucidum are the fibers of the fornix that are coming out of the temporal lobe from the hippocampal formation. Now, the other day I said what we're going to use as a landmark is the interventricular foramen. So that's right here. So the tip of the scissors just went into the interventricular foramen, which means that's a point that means everything caudal to this is diencephalon. So this is all the thalamus, and then at the bottom you can kind of see this widening where the third ventricle would be, and everything down below here is hypothalamus, including this little bump right here, which is the mammillary bodies. That's where the fornix is ultimately going to end. Okay? So my first cut is going to be a horizontal cut. So what I'm going to do is go through using the interventricular foramen and the splenium of the corpus callosum, is I'm going to make a horizontal cut, kind of using those two structures as a landmark. And if you notice, it also is cutting through the rostrum of the corpus callosum anteriorly. Okay? So now, this is our typical view that I showed you as the template I showed you yesterday, in that here we have the internal capsule. Okay, here's this V-shaped structure. This is the anterior part of it, so of the brain. So here's the anterior limb. The genu, or the bend in it, comes right to that interventricular foramen, and then the posterior limb extends caudally. Now, in that V-shaped structure, if you look out lateral to it, you see this large lens-shaped region. So that's the lentiform nucleus, and in this particular brain, doesn't show up in every brain, but in this particular brain, you notice a different color between this lateral most part and this internal most part. This is all putamen out here. All of this internal is globus pallidus. But as a collection, that's all the lentiform nucleus. If we go to the anterior limb of the internal capsule, if we go across it, now we have the head of the caudate nucleus lying within the lateral ventricle. It forms this big bump in the lateral ventricle anteriorly. If we go to the posterior limb of the internal capsule, Now, medial to that and forming the wall of the third ventricle, all of this structure is thalamus. So within that structure, what we can't see is any definition of any thalamic nuclei. So on these things, if I put a pin in there, your answer is thalamus. That's it. Okay? Because I can't tell anything more than that. Now, the other thing, if you look back at where I did this, that cut was through the interventricular foramen, through the splenium of the corpus callosum and the rostrum or genu of the corpus callosum. If we go back to this image here before we go any farther, I also cut right through the anterior cerebral artery coming up like this.
Move the brain forward.
Picky. So,
Could you say that again?
okay, so here on the midline, you can see this arterial branch right here. This is the anterior cerebral artery that's sweeping around corpus callosum, and it's going to come back and end about the parieto-occipital sulcus, which kind of right in here. I don't have it cleared off very well. Okay? So all of this is the anterior cerebral artery blood territory. If we look at this without too much definition from the lateral side, if we follow the singular sulcus, then we have this branch coming up, and this is the marginal branch. This whole region is the midline continuation of primary sensory, primary motor cortex, and this is the region that is representing or contains the lower extremity representation of the homunculus. So the answer to the question, blood supply to the lower extremity part of the homunculus, is by the anterior cerebral artery. Okay? Back here in the occipital lobe, it's the posterior cerebral artery. So then, if we then take this apart and we look at that cut that we have, here's our rostrum of the corpus callosum, splenium of the corpus callosum, and here we have all of this midline structure. We have cortical structure anteriorly and cortical structure posteriorly. The blood supply to this area is the anterior cerebral artery. The blood supply to this area is posterior cerebral artery. Okay? No middle cerebral artery on this midline part. Okay? Now, what I'm going to do, so our other landmark was what? What was our other landmark I talked about yesterday, or whenever it was, Monday? It was specific to the limbic system. So this bump right here is the uncus.
Oh, right.
Okay? So that marks the position of the amygdala. Okay? So if I make a couple more horizontal cuts that ultimately get down to that level, then I am also cutting horizontally through the temporal lobe. And I should be able, if I get to that level, should be able to look at that horizontal cut and see the two structures of the limbic system that lie within the temporal lobe. And that's the amygdala and the hippocampus. Okay? So I'm going to do this in two cuts. This is a pretty stiff brain, so I can do that.
It'll be fried in butter, right?
Yeah.
It's like cauliflower.
So in doing that, in making those cuts, I lost the brainstem and the cerebellum. Okay, but if we look at this on the midline, I have a cut here that then is traveling through the inferior part of the thalamus and the hypothalamus and just going into the midbrain. So I should be able to look in there and possibly see structures of the midbrain, like cerebral peduncle, superior colliculus, red nucleus, substantia nigra. The next cut is inferior, and it's right through the uncus, and kind of running transversely through that temporal lobe. So this is just the last part of the temporal lobe. Okay.
What is the uncus again? I mean, what does it do again?
What does it do? I didn't really tell you what it did. I said it is the position of the amygdala, which is that part of the limbic system that's taking sensory information and tagging it to emotional pieces. In actuality, the cortex that's on the surface of it, okay? But I didn't go there. So now what I'm going to do is, I want to take, if we look at this image, this midline image, and see where I made that cut, we can see how it's just starting to go through the beginning of, or the most rostral part of the midbrain. So I pull that away, and now we look at this. And if you look at this, you notice that there's a dark circle right here. It's your hand. Okay, so right here is the red nucleus. This is the superior colliculus. And you can see these striated fibers right here along the side. And that's where the internal capsule is starting to come down and collect and become the cerebral peduncles. Okay? Remember I showed you yesterday or Monday that there are fibers from all parts of the cortex that are ultimately coming in to form that cerebral peduncle. They're destined for the pons. They're destined for brainstem cranial nerve nuclei, and they're destined for the spine. Okay?
Can you show the red nucleus and the superior colliculus again?
This is the red nucleus right here, and this bump is the superior colliculus. Okay? Now, the next cut then just takes off that bottommost part of the temporal lobe, but if I hold this back in place so we can see it, right there's the uncus. There's that bump. Okay? So if I remove that section, now we're looking at the temporal lobe in a horizontal. What? Now we see, so we can see this bump that was the uncus. Okay, that's the bump. Inside that we see, here's the very tip of the lateral ventricle right here. Anterior to that lateral ventricle is this area, this homogeneous looking area of gray matter, is the amygdala. Behind it, and on the other side of the ventricle, you can see now there's sort of this kind of squiggly looking structure here. That is the beginning of the hippocampal formation. If we look at this previous section, let's see if I can do this not easily. So let me flip this over. So now we're looking at, you know, if we did this, this is the same as just a mirror image of that. Here's the uncus, here's the amygdala, and we can see this white matter squiggle through here. And those are the fibers that are coming out of the hippocampus. The hippocampus is folded inside of the parahippocampal gyrus. If we come back up to that previous level, now we can see kind of this. It's not a great one, but we can see kind of this structure right here. And this is the caudalmost end of the hippocampal formation. So it lies folded inside of that parahippocampal gyrus all the way along until it reaches the corpus callosum. And that's about the end of it. And from there, it's just the axons, those efferent axons forming the fornix. that continue all the way back around to the mammillary bodies, okay? So that, from this point of view, what you need to think about is, if I look at this, can I put this all back together in some sort of area that I can then look at this on the midline and kind of imagine the orientation of those nuclei. Now, what I want to do is I'm going to take everything away that I'm not going to use for the next one, okay? Which is, I just want to be able to look at this image that defines best the anterior genu, posterior limb of the internal capsule, caudate, lentiform nucleus, and thalamus, okay? And there's a reason I want to do that, okay? Because now I'm going to take the other hemisphere, and I'm going to use the exact same landmarks that I used before. Here's the interventricular foramen. Here's corpus callosum, septum pellucidum, the membrane between the two lateral ventricles is on this hemisphere primarily, but you can see a little remnant of it here. And I'm going to take this interventricular foramen, and I'm going to take the uncus of this side, and I'm going to use that as a landmark. So here's the uncus right here, the bump of periapicampal gyrus. I'm going to take and I'm going to make a coronal cut, just like that. Okay? Now, if we look at that, and we look at that with respect to putting it at that same level. Okay?
Pause.
What?
Pause.
Time out.
Jamie's messing with the camera. Oh, shoot, now I'm.
It's okay, we got 4 hours.
I should be Facebook Live.
That was
this.
positive.
That'd be genius. My friends would be like,
Oh, good point.
yeah, you guys are now. You're like, "Sure, we can stay till 5. It's fine.
Yeah.
You okay, Jamie? No.
We're almost there.
Okay.
And play. Okay.
It's the best way to do it.
So what I've done as a point of reference is I've taken that horizontal section, that's that template that we've used with respect to the interventricular foramen, and placed it next to its matching hemisphere. So now I have the coronal view of that same level. In other words, if I took and went across in a coronal fashion like this, on this image, I should be able to see internal capsule, lentiform nucleus, insula, lateral sulcus, and the temporal lobe. So if I do that same thing on this side, interventricular foramen, internal capsule, lentiform nucleus, insula, lateral sulcus, temporal lobe. So as a way of orienting yourself, that's kind of what you see crossing in that way, okay? Now, when we look at this, if we look at this on the midline, the reason we haven't defined any thalamus is because the thalamus doesn't go any farther anterior than that interventricular foramen. So we don't have any thalamus on this. All we have is we have the caudate up here, and we have lentiform nucleus down here, and we have internal capsule separating them. Okay? So now what I'm going to do is I'm going to make a more posterior coronal cut. And before I separate it, what I want you to look at, where that cut is, now it's back here, it's going through the corpus callosum, through the fornix, now it's going through the thalamus, but now it's this coronal cut that's cutting through the midbrain and the ventral pons. Okay? So if I open that up, here's corpus callosum. Here is the internal capsule coming up. All of this is thalamus. And because of this caudal cut, so if I make this, do this same thing, if I put it together in that format, if I cut through here, I'm going to see thalamus, and then I'm just going to get the tail end of the lentiform nucleus as it crosses that posterior limb. Then I'm going to get insula, temporal lobe. Okay, same thing. So here I'm coming across the thalamus, I cross the internal capsule, and then I get this little tiny bit of the lentiform nucleus, then the insula, lateral sulcus, and all the temporal lobe. Okay? So now let me put this back together. Okay, so remember that I made my landmarks were the interventricular foramen right here and the uncus. Now in this case we're anterior, this cuts clear out here because we were coronal through the uncus. So here's the uncus, and now we see this large area beneath it, deep to it, and all of that is the amygdala. Okay? If we make a cut more caudal to that, now we jump behind the ventricle, or to the ventricle, and now we're in the hippocampal formation. So if I do that, and I look at this, now what we see is in that same medial part of temporal lobe, here we see this C-shaped structure right here. And that's the hippocampal formation. Okay? If we look at this is a little hard because of some of the blood that's in this brain because of the strokes most likely, is if we look at this, here is, if we look on this side, here's our interventricular foramen coming into fourth ventricle. Okay? So if we look at that, so up here's thalamus, down here we're starting to go into the midbrain, and then here we're going into the pons. So if we look at the midbrain part of this, from here to here we have the posterior limb of the internal capsule becoming continuous with these fibers right here that are on the lateral side of the midbrain. This is cerebral peduncle. If we come just medial to that, we see this blackened line of cells right here. Substantia nigra pars compacta. Okay? If we look on the midline again to get sort of this orientation, and we relate that, here's superior colliculus, inferior colliculus, so our midbrain's going to end right about here, and now all of this is pons, and all we're cutting through is the ventral pons, so we can just see this structure, and we can see it forming the middle cerebellar peduncle off to the side. Okay? So I'm going to make one more cut. So I'll put it back together again. From that interventricular foramen, and look where that last cut is. So still cutting through the midbrain up here, but now we're cutting through the tegmentum of the pons, and now we're cutting into the medulla. Okay? So if we look at that, so caudal thalamus right here, nothing in terms of the lentiform nucleus anymore. We can still see that C-shaped structure of the hippocampus, but now we're at the superior colliculus. Here's the cerebral aqueduct going through, and now this is the tegmentum of the pons and the cerebellar peduncle, middle cerebellar peduncle, and now we're down into the medulla. Okay? So when you look at, when you put this back together and you look at it kind of on the midline and try to relate those areas, so on the midline, the pons stands out because of the bump, but everything that extends caudal to that is going to be medulla, or if it was long enough, and this one was a short one, would it even go on down into the spinal cord? Because a lot of those brains we took out, kind of cut the spinal cord to C3, C4, and used that as a tail to pull it up. This one just happened to be a shorter one, okay? So the idea of this exercise is to then become comfortable kind of looking at these sections and saying, can I look at this and identify those deep structures? And again, in this image, or even in this one, you can kind of tell the difference between putamen and globus pallidus. That's just hung on by the arachnoid, and it will eventually fall away. But on this one, this side, you can't really see any different look to lateral versus medial. In other words, you can't differentiate putamen from globus pallidus on this one, okay, on this side. Can on that side, probably because some of that bleeding from these hemorrhages has kind of invaded that some way, okay? Probably striatal arteries of some sort, all right? But ultimately the idea is, can you go back and look at all these structures and say, if I identify it, if I can identify the amygdala here or here, can I just do that? Can I say that's the amygdala? Can I say what the function of it is? If it were pinned in a brain section, or it was pinned in that slide 10, or an arrow on that slide 10, which is the medial part of the temporal lobe, and it says, what's its function? Just attaching emotion to sensation or something like that, okay? So that's kind of the process to go through. There are a lot more things you can find in the handout.